Okay, so welcome back to this next video in which we are discussing ligand-gated ion channels. Okay, so we've seen how there are three major families of ligand-gated ion channels. There is the P2X receptor-like family, there's the glutamate receptor-like family, and then there's the nicotine receptor-like family, which is saying it means all the receptors which are similar to the nicotinic acetylcholine receptor, which is the receptor that is sensitive to nicotine. Okay, and we'll study this receptor in a lot more detail and see how nicotine exerts its effect. Okay, so we've seen this one example of the nicotinic acetylcholine receptor, which is within this family, which is also known as the cis-loop ligand-gated ion channels because of their cis loop on the extracellular aspect of the protein subunits which make up these channels. Okay, but now let's see another example, which is a receptor that responds to 5-HT, also known as serotonin. So I think I'll get another piece of paper for this because we don't want to squash it in on that little bit there. Okay, right, so let's now talk about 5-HT. Okay, so 5-HT stands for 5-hydroxytryptamine. 5-hydroxytryptamine, okay, and it's also known as serotonin. Now, serotonin is a very famous neurotransmitter because of its involvement in mood, basically. It's the neurotransmitter system which is targeted by the SSRIs, the selective serotonin reuptake inhibitors, which are a very common now treatment for depression. Okay, so for that reason, it's very famous. The drug, the infamous drug Prozac, proper name fluoxetine, works by interacting with serotonin, by increasing the levels of serotonin within the brain. Okay, right, so let's see the structure of serotonin, and then we'll look at the receptors to serotonin, specifically the cis-loop ligand-gated ion channel receptors to serotonin. Okay, so the structure of serotonin, it's basically made from the amino acid tryptophan. So I'll firstly show you the amino acid tryptophan and then show you how 5-hydroxytryptamine is basically just a modification of tryptophan. So, okay, so let's start with the core amino acid structure. So here's the amino acid, well, here's the amino group rather. Here's the alpha carbon of the amino group. Here's the carboxylic acid group, down here off the alpha carbon. Okay, and then the R group of tryptophan is that you have a methylene group, like so. Okay, and then you have a structure known as a indole ring. So I need to tell you what an indole ring is, but before I can tell you what an indole ring is, I need to tell you what a pyrrole ring is, because an indole ring is a modified pyrrole ring. So let me show you what a pyrrole ring is. A pyrrole ring is a type of aromatic ring which has 5 members. Now 4 of these are carbons, as shown here, and then the 5th one is a nitrogen. Okay, now let's put in the double bonds. So you have a double bond here and a double bond here, and the whole structure is beautifully symmetric. You then just stick hydrogens off every single member of this ring, like so, and that gives you the pyrrole ring. Okay, now let's discuss the indole ring, which is basically a modified version of the pyrrole ring. It's a pyrrole ring with another ring stuck off it. Okay, so the indole ring actually has 2 rings, really. So let's start off by recopying our pyrrole ring out. So our pyrrole ring is this 5-membered ring here. Okay, and then it's got a 6-membered ring coming off it, which is all carbons. Okay, so it's effectively a benzene ring coming off it. So here we go, and let's put in the double bonds for the pyrrole ring here. Now let's put in the other double bonds for benzene. So remember, benzene has alternating single and double bonds. And now then stick hydrogens everywhere that you need a hydrogen. So here's a hydrogen, and then hydrogens off all these other carbons in the benzene ring. So one off here, and then one off here. Okay, that's the structure of an indole ring. We're now just going to stick that on here, basically. And we're going to take this hydrogen off here and stick the whole thing over there. So we're going to put an indole ring on the side of this methylene group. So here goes. Okay, so here's the pyrrole ring initially. Here's the nitrogen of the pyrrole ring. And then here is the benzene ring with alternating single and double bonds. Okay, and that forms us the full indole ring here. Nitrogen here with a hydrogen off. Hydrogen, hydrogen, hydrogen. Another hydrogen off there, and then a hydrogen off here. So, this now is the structure of the amino acid tryptophan. Okay, and it's from tryptophan that we get 5-hydroxytryptamine. Okay, so tryptophan has the single letter amino acid code W, just to add a bit of extra knowledge into this. So, how do we go from tryptophan to 5-hydroxytryptamine? So basically, the way that you go from tryptophan to tryptamine is that you take off the carboxylic acid group here and replace it by a hydrogen atom. So let's start off with that conversion, the removal of the carboxylic acid group. Okay, so here's the amino group here. Okay, here's the alpha carbon with a hydrogen off it. And now, instead of a carboxylic acid group here, instead you've got just a hydrogen. Then you've got the methylene group of the R group. And then you've got the indole ring coming off that. So here is the indole ring, this 5-membered pyrrole ring starting us off, okay, and then the benzene ring off that pyrrole ring, like so. Okay, and now all that we have to do now is just add a few hydrogens on wherever we need one. So hydrogen here, and hydrogens off all of these 4 carbons within the benzene ring here. Okay, right, so that creates us tryptamine. Oh, sorry, tryptamine. Yes, that's better. Tryptamine. Okay, so you don't use the O anymore, you replace it with the amine, tryptamine. Okay, and that's because it's got the amino group up here, and no longer has it got the carboxylic acid group. Okay, and then finally what we want is 5-hydroxytryptamine. Now, for that, we know we just need to add a hydroxy group on. The difficulty is which carbon is the 5th carbon. So I need to teach you how we label the members of the tryptophan, or the tryptamine molecule here. So, how do you label these members? Well, basically, it starts off nice and simple. We call the old alpha carbon of the amino acid, we continue to call that the alpha carbon. So this is still the alpha carbon. This is the beta carbon that comes off the alpha carbon. Okay, nice and simple. Now we abandon that naming system and start a new naming system for the indole ring here. Okay, so we call this nitrogen here, since it's the only special element in this entire structure, we should call it 1, basically. So we start off with this nitrogen here. Now, we can go either way after this. We want basically to go round rotationally. Okay? Now, we could go this way, or we could go this way. But we go this way, basically. We go round like this and finish up back here. So, we start with the pyrrole ring. So we go to this one next, and we call that the 2nd member. Then we go here, we call that the 3rd member. We go here, and we don't call that the 4th member. We call it 3A instead. Okay? We then go here, we call that the 4th. We go here, we call this the 5th. Okay, so we're done, basically. We know where the 5th is now. We call this one the 6th, this one 7th, and then we have to stick to this silliness up here where we called this 3A. So we also call this 7A here. Right, so 1, 2, 3, 3A, 4, 5, 6, 7, 7A, basically, is how you name the members of the indole ring in tryptamine. So now we know exactly where we need to add this hydroxyl group onto tryptamine to make 5-hydroxytryptamine, or serotonin. So let me show you this. Okay, so finally, just to finish the whole thing off, here's the alpha carbon. Here's the amino group off the alpha carbon. And you still have these 2 hydrogens off that alpha carbon. You then have the beta carbon in this methylene group here. Okay, and then you have the pyrrole ring here. Okay, so here's our, whoops, can you see this? Yes, I'll bring it up. Okay, so here's our pyrrole ring, this 5-membered ring where 4 of the members are carbons and then 1 is a nitrogen. And now we've got this benzene ring off the side of the pyrrole ring, turning this whole structure into an indole ring rather than a pyrrole ring. Okay, and then we need to add a hydroxyl group onto this 5th carbon here. So remember the way we named it. 1, 2, 3, 3A, 4, 5. So here is the hydroxyl group of our 5th carbon, and now we just stick hydrogens onto anything else that needs it. Hydrogen here, hydrogen here, a hydrogen here. We don't need a hydrogen off the 5th carbon anymore, but we do need a hydrogen off this 4th carbon. Okay, and this is 5-hydroxytryptamine, or 5-HT for short, or serotonin is its more popular name. Okay, right. So, serotonin receptors then, 5-HT receptors, most of them are not ligand-gated ion channels. So remember when we discussed the 4 different types of receptors, we had ligand-gated ion channels was a type of receptor here. But we also had G-protein coupled receptors, receptor tyrosine kinase, and nuclear receptors. Now, most serotonin receptors are G-protein coupled receptors. However, there are a type of serotonin receptors which are ligand-gated ion channels, and these are the 5-HT free receptors. So if ever you're talking about 5-HT receptors, then basically they're named in the order they were discovered, basically. So there's 5-HT1 receptors, 5-HT2 receptors, and then you get to 5-HT3 receptors. Now both the first type of receptor and the second type of receptor, they are both G-protein coupled receptors. But the third type is a ligand-gated ion channel. And I also want to stress that in these divisions, there is not just one type of receptor. So 5-HT3 receptors are basically a whole class of receptors. It's not just one receptor, it's a family of receptors that are ligand-gated ion channels of the cis-loop ligand-gated ion channel family, which respond to serotonin. Okay, so serotonin binds to this receptor, which is a pentameric receptor of subunits which have this cis-loop structure. And when it binds, the channel opens, and what does it allow through? Does it allow cations or anions? Well, it, like the nicotinic acetylcholine receptor, allows cations through, and therefore is going to produce you an excitatory postsynaptic potential. So basically, this receptor, 5-HT, acting on 5-HT3 receptors, is excitatory. It's going to increase the likelihood that the postsynaptic cell undergoes an action potential. Okay, so that will do for this video. In the next video, we'll look at 2 further examples of cis-loop ligand-gated ion channels.